All right, so we are going to be working on test corrections. That's what this video is for. You will need to answer these questions correctly on the quizzes. I'm sorry, not the quizzes, but the video in order to receive points back for these questions. Now, of course, you're only going to be able to earn up to half points back of what they were originally worth. If you miss the question after it's been explained to you, you do not earn those points back. Okay, so let's take a look at number one. It says a student was preparing for a hike and wants to pack a snack that has biomolecules that provide quickly available energy but few excess calories. Which nutrition label lists the best combination of the biomolecules that provide quickly available energy while providing fewest calories from other types of biomolecules? Okay, so what are we looking for? We are looking for quick energy, but few calories. All right. Now, one of the things that we need to remember here is what are the biomolecules that we can eat for energy? We can eat carbs, we can eat lipids, whoops, and we can eat proteins. Okay. Now, when we eat carbohydrates, for every 1 gram, we get 4 calories. Lipids, for every 1 gram, we get 9 calories. And for proteins, for every 1 gram, we get 4 calories. So we are looking for something that is high in carbohydrates or proteins, not lipids. All right? Because we want fast energy. Now, when we're talking about fast energy, we are talking about our carbohydrates, lipids, long term, and proteins are a secondary energy. Okay? So let's take a look at our answer choices. All right, I realize that this is kind of hard to see, but let's go with me on this because my printer was running out of ink. So here we've got 20 calories. All right, 0 fat, which is lipids. Calories. 0 calories. It's carbs. 0 carbohydrates. And 5 grams protein. All right. On B, we have 180 calories. All right. Then we are going to have fat, 8 grams of fat. Carbohydrates, 13 grams of carbs. And 15 grams of protein. All right. Next, we have 975 calories. Already, remember, this is already too much. Remember, we want few calories. So we're not going to even calculate this one. Okay? So let's take a look at this one. We have 140 calories. Now, just by looking at 140 calories, we know that we can eliminate this one because this one and this one are going to be the smallest ones. Okay? We have 4 grams of fat. All right. 22 grams carbs and 5 grams of protein. Now, let's take a look at, remember what we were looking for. We were looking for quick energy, few calories. Our fast energy always comes from our carbohydrates. So your answer here, it's not going to be A because this is not fast energy. This is a secondary source. Your answer is going to be D. Okay? Now let's look at question number 2. The complex carbohydrate, hold on, let me fix the zoom. The complex carbohydrates pictured below are made by linking molecules of glucose. In all three complex carbohydrates, the subunits of glucose are bonded together differently because they have different structures. That means they most likely what? Your key word here is different. Okay? Now, we know that we are dealing with a carbohydrate with a monomer that's glucose. Okay, so all of these molecules here, glucose, 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 and together they all make a complex carbohydrate, a polysaccharide. Now, what's the correct answer here? Remember that if they have different structure, okay, shape determines function. All right. It says they contain different chemical elements. All carbohydrates have carbon, hydrogen, and oxygen. So it can't be that they have different elements. They also don't make proteins. Carbohydrates, these are sugars. Okay. Carbohydrates, sugars. All right. They don't have nucleotides, but they do perform different functions. So even though these are all carbohydrates, okay, they're all polysaccharides, the shape determines function. This one right here, stored sugar in plants. Okay, cellulose, cell wall in plants. Glycogen, stored sugar in animals. So your answer here was J. They perform different functions. Okay. All right. Our next one is number 4. Now, the questions that we're doing there are just the most missed questions. The human body uses specialized proteins to speed up chemical reactions. These proteins are called enzymes. Without enzymes, these reactions in the body would happen too slowly to keep you alive. Enzymes are made during protein synthesis, which occurs at the cell's ribosome. Which biomolecule performs the instructions to build enzymes? I'm going to tell you a secret. There's nothing in here that would help you answer this question properly. Nothing. It all has to do with here. The instructions, genetic code. Okay. Blueprint for cells. Your traits, characteristics. The instructions are going to be DNA, okay, which is nucleic acid, that NA, nucleic acid. All right, moving on to our next question, which is number 8. Lactose is a sugar found in dairy products. The body converts it into a molecule that can be used to make ATP energy. All we cared about right here, just energy. What type of biomolecule is lactose? Sugar. Ose. Sugar. What are all sugars? All sugars are carbohydrates, okay? Every time. None of those other ones can be any other thing. Lipids, fats, nucleic acid, genetic material, proteins have many different kinds of functions, but the only one that is ever sugar is always carbohydrates. This one is one of the only questions I'm disappointed that this was most missed. All right? Don't know how. I don't know if it's just not paying attention or what's going on here, but this question should have never been missed. All right, our next question is number 9. A model of the cell membrane shows many different molecules that perform various functions for the cell. Some of these molecules keep the contents inside of the cell separated from the contents outside. Others allow certain materials to be transported across the cell membrane. Which molecule separates the contents of inside of the cell from the contents outside of the cell? What is this molecule? It's the cell membrane is probably 80 plus percent lipids and probably 20% or less protein. Which one of these do you think it's going to be? It's going to be the lipids. Look, all of these are lipids, okay? They're helping to protect the inside of the cell. Okay? So your answer here was lipids. All right, another thing. This question, almost everyone got this one correct. Guys, you have to look for your context clues here. Here's protein, carbs, lipids, and nucleic acids. Okay? Make sure you're using all of the clues that are available to you. Don't be in such a rush to get it done that you're missing important things. All right. Carbohydrates and lipids both serve as a source of energy for organisms. What kind of energy do carbohydrates and lipids provide? So, carbs and lipids are a source of energy. Carbohydrates are the ones that your body will burn faster. That's why they're a quick source of energy. Lipids, it even starts with an L. Think lipids long term. Okay? Lipids long term. All right. Oops, hold on. Missed a question. All right. Number 14. Lactose is found in milk products. It is converted by the body into a usable form in a series of chemical reactions. The diagram shows the series of reactions that convert lactose into a usable form. So here is the chemical reaction. All right. Okay. If enzyme 2 is denatured, what level of substances will increase? Remember, denatured means that it is not functioning. FUNCTI. Okay, not working. I feel like I spelled that wrong, but we're just gonna go with it. So we're gonna go broken. Okay. So that means if this one was taken out, What substance will increase more? So, what substance do you have more of? So, enzyme 1 breaks this. All right. Enzyme 2 breaks this. So, what are you going to have more of? You're going to have more of galactose. Okay. Okay, now here we have number 15. Okay, the diagram represents one way an enzyme can be inhibited. All right, so to be inhibited means to prevent something from happening. All right, so which statement explains the effect on the inhibitor on the enzyme? So here is normal, and here is the enzyme inhibitor. This is what they are asking about. This is your question. So, what's changed from here to here? You have this inhibitor. Can the substrate bind there? No, it cannot. It's going to move on. Can't bind. Okay. So, your answer here is D. The substrate will be unable to attach to the enzyme because the shape of active site has changed. Okay. Shape of active site is changed. Substrate won't fit. All right. All right. The enzyme substrate complex can be modeled using these 3 steps. Okay. What does step 3 illustrate in the diagram? So they're asking about this one in particular. First, in order to do that, we need to identify some things. Y is the enzyme. Okay. Now, in order for the enzyme to work, a substrate has to bind to it. And what gets made is the products. Okay, so your answer here was the conversion of substrates into products. They're only asking about this one right here. Okay, all right, moving on. Amylase is a digestive enzyme in the mouth that increases the rate of reaction for starches breaking down into simple sugars. The mouth's environment is close to neutral with a pH near 7. Okay. When amylase is exposed to acid in the stomach, the pH range near 2, the enzyme is denatured. All right. What graph best demonstrates the rate of reaction caused by amylase? Okay, look. Here was your key right here. At the stomach, pH near 2, the enzyme is denatured. This means that no rate of reaction. Okay, that means the enzyme is not working. No enzyme function. Okay, but it's working best. Amylase is in the mouth. Okay, the mouth's environment is at pH 7. Okay, so pH 7 is good, best, optimal. Okay, pH 2, bad, denatured. Not working. All right, so let's take a look. Does this show that the rate of reaction is high? Okay, so here is 7. Is this a high rate of reaction? Remember, your rate of reaction is going to increase. Okay, so this is no. Number 8, pH 7. Do you have the high rate of reaction there? No, you don't. pH 7. Again, you have a decreasing rate of reaction. pH 7. Look, optimal. Highest rate of reaction right here. Additionally, we can look at pH 2. At pH 2, we have no reaction, which is good, but we have a lot of reaction here at pH 2. That's pH 2 is bad. All right? Look, no reaction here at pH 2. Your answer is D. All right. Amylase is an enzyme that helps break down complex carbohydrates into simple carbohydrates. This enzyme is present in the saliva of the mouth. Again, this is from question 19. All right. When salivary amylase is in the stomach, it stops functioning. Which of the following best explains the change that occurs when it enters the stomach? So it's in the mouth. Amylase is an enzyme. So think about your digestion, okay? When amylase is in the stomach, it stops functioning. Your stomach is acidic. All right? Stops functioning means denatured. So amylase is used up in the digestion of proteins in the stomach. The alkaline pH of the stomach inhibits the amylase. Amylase is denatured in the acidic pH of the stomach. Hello, we should know this one. And it's definitely not all complex carbohydrates are already broken down when swallowed because, guys, you have to use the clues that are in these questions, okay? Number 21. What type of substance serves as a catalyst, okay, for reactions that break large food molecules into smaller? Break large molecules into smaller, okay? A catalyst. What's another word for a catalyst? Is an enzyme. Also known as enzymes, okay? So A. All right. Number 24. This was a pre-AP question. So 23, 24, all of that from here on is a pre-AP question. So this is extra. So for the level biology students, guys, just pay attention and I'm going to help you get this one. What's function of the protein found in the cell membrane? All right, hold on just a moment. Let me get the cell membrane picture from earlier. Hush, Sadie. Okay, here is an image of the cell membrane. Okay, so these right here, you have a lipid bilayer, and these things right here are proteins. Now, one of the functions of these proteins is to transport molecules in and out of the cell. So your answer here was F, to facilitate the movement of particles across the cell membrane. All right, so I hope that this was helpful. I hope that you feel more confident in the information. If you are still stuck, come to tutorials, okay? So,